Hi, in this first lecture we are going to talk about complex systems. It's now becoming clear that in many sciences we face very similar issues. It's not only because we are dealing with large datasets, it's also because complex systems in general share common design principles. In systems biology, we mostly care about the design principles of cells. In this course we'll deal mostly with mammalian cells. However, we'll try to identify analogies between human cells and technological complex systems. So what is science? One definition is systematic knowledge of the physical or material world gained through observations and experimentations. So one thing to keep in mind about observations is that they have to be reproducible. Once we have a reproducible observation, we can form generalizations. Generalizations can be formulated mathematically or programmatically into models. These models give rise to theories. Once we have an accurate model, we can perform simulations or computations that can be used to predict unseen yet observations. The question about models and theories is how accurate and general they are. Once we obtain systematic knowledge about the physical and material world, we can use this knowledge to create new tools. Here, innovation and creativity are critical, and these new tools can now compete in an economic or in a social market, and the most fit tools will survive and evolve. So let's think of an example. One obvious example is the circular rhythms of days, months, years. These observations are well understood and can be predicted. Then, using this knowledge, calendars, clocks, and tools were invented by humans based on this scientific understanding. So one thing I would like you to try is to think about other examples. If you are coming from the fields of biology or systems biology, I would like you to think of examples where scientific discoveries led to the development of tools that helped us advance scientific discovery, further scientific discovery in biology. If you come in from other fields, I'm sure you can find examples where science was used to develop new tools that were based on that science. So there is a yin-yang relationship or feedback loop between technology and science. So through technology that was assisted by science, we produce new tools that can be better used to observe things, and that advances science. Technology is also producing new complex systems that can be studied by science, for example, the stock market or the social network on Facebook. So far, almost all complex systems that were created by humans that have technological origins are created to extend human fitness. So those are tools that we invented to extend our fitness. So what is a complex system? Complex systems are made of many heterogeneous, diverse agents or agent parts that interact in various ways to form higher order entities. Complex systems are networks of agents. Agents are made of other agents or made from agent parts. However, complex systems are relatively difficult to define because they contain many properties. So there's no one characteristic that makes a complex system a complex system. So what is the science of complexity? So in the 80s, a set of prolific scientists banded together to form this new science, or to define this new science, to try to identify concepts across sciences. So the goal was to better understand, generalize, theorize, and predict the behavior of complex systems in general, not just in a particular field. So I use the term agent when I defined complex systems. So what is an agent exactly? And an agent is a term that was invented by those complex system researchers, and it's an entity in complex system. It can actually be a complex system on its own, and it contains properties such as actuators. Those could be our arms and legs if the complex system is a human. Complex systems typically contain sensors, and these are our eyes or our nose, for example, if we are consider ourselves as a complex system. Typically, complex systems have a defense system. This could be our immune system in our body, or it could be some type of a mechanism within cells to actually have a self-defense from external attacks. Agents typically consume energy, and so they seek energy sources, consume them, convert them to an energy that can be utilized to do the various tasks that a complex agent is doing. Agents typically have boundaries, some type of separator, a wall that separates the agents from other agents, as well as separate the agents from the environment. Natural agents can have this amazing ability to self-reproduce. Technological agents are typically reproduced in factories. Agents typically have some type of an oscillator inside of them, such as our heart or a circadian rhythm clock in cells. Many agents have a central processing unit. This could be our brain, or in the case of a cell, those are those networks that we're going to talk about, those cell signaling and gene regulatory networks that control the behavior of a cell. Agents also typically have a ability to self-repair. And if we think about cities, for example, this could be the police, hospitals. Agents also have advanced transportation systems. One thing to keep in mind about natural biological complex systems is that they appear at different scales. So at the subcellular scale, we have complex agents such as the nucleus or the mitochondria, and those are independent systems within the system of a cell. The next level is the human cell, and that's going to be the focus of our course. Then the next level would be tissues and cell populations. This could be a heart, a liver, or a bacterial colony. Multicellular organisms such as us is another level of a scale in organization of natural complex systems. And the final level is those are ecosystems, social structures such as a pond, a rainforest, a beehive, countries, companies, and families. So one thing that I would like you to try is to identify at every one of those scales what are the agent component or the complex systems properties, such as the actuators, the sensors, in each of those systems that I mentioned here at the different layers of scale. So I mentioned that technology extends human fitness. So humans have invented products that increase their fitness. For example, a motorcycle increases our ability to move. Sunglasses or a microscope help us to improve our senses. Clothes help our defense from the natural environment. A megaphone or cell phones help us to communicate. And computers help us in general to process information and do a lot more things. So when I mentioned complex systems, I said that there are very different types of complex systems, and sometimes they share their properties and sometimes they don't. We have to distinguish between natural evolution and technological evolution. And there are differences between those types of systems that emerge in natural evolution and man-made evolution. And some of the differences are that the natural evolution takes a long time, billions of years to evolve, while man-made evolution seems to accelerate, and it's only been accelerating and on the move for maybe thousands of years. So one is very slow and the other one is fast. Natural evolution is limited to mutations that have to pass through generations. Whereas in technological evolution, you can have an idea and that can become a product overnight. However, there's some similarity between natural and man-made evolution. For example, the rate of evolution is different across different parts of our planet. In some areas, such as major cities, you see rapid evolution, while in other areas there is very slow adaptation of change. We also want to distinguish between open and closed complex systems. So what I mean by closed systems, I'm talking about an individual agent that can be a complex system, like I mentioned before. So closed systems have defined boundaries, a central processing unit, a clock, sensors, actuators, utility apparatus. They are born, live, and die. They only grow to a defined size, they self-repair and reproduce. Open systems, on the other hand, have less defined boundaries, not clear governance. They have many agents of the same type, like the beehive or an ant colony, all acting stochastically, meaning that they act in random fashion. However, together they lead to an emergent behavior. So they look like they're all working together, even though individually they are acting randomly or self-serving. Open systems grow faster as they become more complex and diverse, and this is a general assessment. On the other hand, closed systems become less flexible as they grow in complexity. So it's harder for them to change as complexity increases. And this is an example. One ant is a closed system, but an ant colony can be considered an open system. I would like you to identify from your own personal experience some open and closed systems that you can consider complex systems, man-made and natural. So these are some examples to get you started. So the human cell, obviously, or human, a cow, the weather system, stock market, New York City, a tree, commercial air traffic systems. So not only try to identify additional complex systems, but also try to classify them as whether they are closed or open. So now I would like to focus on what this course is about, and this is the human or mouse cell. And here, after listening to these ideas about complex systems and agents, let's try to identify the parts of a cell. So now let's think about the human or mouse cell as an agent. Let's try to identify in the cell those general properties of agents in complex systems. So if we think about actuators, those could be decisions made by a cell, such as to crawl towards a specific direction, to differentiate into a specific lineage, to perform self-execution, which is apoptosis, or to proliferate. When we talk about sensors, and we're going to talk about those more later on, these could be receptors at the cell membrane or receptors at the nucleus. Those proteins listen to the environment and transmit information about the status of the environment into the insides of the cell. Cells also have defense systems, such as the interferon system. Cells have several boundaries, for example, the plasma membrane or the nuclear membrane. Cells obviously consume energy, for example, through conversion of glucose into ATP. Cells can self-reproduce, like I mentioned, through a cell cycle. They have a clock, which is typically the circadian rhythm clock. They have various transportation systems, for example, the retrograde transport, vesicle transports mechanisms. Cells also have self-repair mechanisms such as the proteasome or the autophagy system. And the central processing system in a cell could be considered the cell signaling network and the gene regulatory network, which would be the main focus of our course. So I know that many of you are not coming from the field of systems biology, so you may not be familiar with the nomenclature. So one thing that I decided to do, I decided to have some introduction to some of the most important concepts that you need to really grasp before you starting to apply those network analysis method to the biological system that is the human or mouse cell. However, I did not want to bore you with some list of definitions, because in biology a lot of it is nomenclature, learning a language that describes different things. On YouTube there's those amazing animated movies that people have posted to visualize what we believe is happening inside a cell or inside our body. So I took pieces, slices from those YouTube movies and organized them to show you some of the most important concepts of molecular and cell biology as we understand it today. And this would sort of like just give you a taste, the type of mysterious and exciting and amazing things that we are discovering that are happening at the molecular level inside our cells. You also would appreciate the complexity of the problems that we're trying to address. One thing that we're going to also do after each of those movies or videos that we're going to watch, I'm going to ask you some questions about some of the most important concepts that I would like you to take from those specific movies. So in some cases you may have to watch those movies once or twice to just make sure that you remember some of the most important concepts that then will be helpful when we applying our methods to analyze those networks that are making the human and mouse cell. So the first movie doesn't give you too much new information, but it really motivates you to think about the fact that we are made of cells, and those cells are living in our body and functioning to keep us alive. Human body is made of arguably between 200 and 400 different cell types, but there are many copies of those cell types, and all of them are organized very precisely to give rise to tissues and organs, and then to give rise to our full human body.
I don't ever slow up, no I don't take shit. I got no love for the fakeness. If you wanna play tough and wanna hate this, I'll always show up. I don't ever slow up, no I don't take shit. I got no love for the fakeness. If you wanna play tough and wanna hate this, I'll always show up and make a statement. I don't ever slow up, no I don't take shit. I got no love for the fakeness. If you wanna play tough and wanna hate this, I'll always show up and make a statement. Everything I do so instinctive and so passionate. Every word I move so descriptive like an adjective. I got a vendetta against people who patented being negative when you should be getting after it.
I got facts over tracks, this and that. Spitting slow, spitting fast, I could roast, I could gas. Think I'm okay at last, but I don't know if that can erase all the past. And the pettiness, a reflection of the emptiness. Hilarious, you think you're worth my time, you're delirious. Mysterious, because you hide behind a fake exterior. Inferior, you know I'll always be a bit superior. Get off of me, this ain't no humble brag. I want you to hear words, you can say them back. I want you to feel free from the chains that last, and to believe in what you got, it was built to last, yeah. Now that I've been put through hell, I never got anyone's help. I had to do it all myself. I don't ever slow up, no I don't take shit. I got no love for the fakeness. If you wanna play tough and wanna hate this, I'll always show up and make a statement. I don't ever slow up, no I don't take shit. I got no love for the fakeness. If you wanna play tough and wanna hate this, I'll always show up and make a statement. Gonna learn the consequence of being incompetent. Mental health is confidence, dreams and some modestness. I'm not here to save the day, that's for you to take away. I could play a million mind games, but instead of say something not illogical, something that is tactical. Rub it on and watch it go. Make yourself unstoppable. Dreams are irresponsible, but they're always possible. If you just believe, you could be so remarkable. Thoughts in my head, a collage and they spread. I'll be great one day, going off of my meds. No, I'm not giving up, no, I'm not giving in. I will make it to the top, taking off in the wind. I gotta make it, I'm saving every day to taste it. I'm patient, but my mind, it can hardly take it. I'm chasing a dream that I've had for
Another very important topic in this field is the concept of stem cells, and this is a very hot topic in molecular cell biology these days, and it really helps you to understand the concepts of a system view of the human body, how cells make those decisions to become what they can become. And this also has many new exciting medical applications that are foreseen to be implemented in the relatively near future.
At the very earliest stages of development, all cells are equally capable of giving rise to an entire organism. But after only a few rounds of cell division, the cells begin to lose this totipotency and to differentiate, sorting into groups that will either contribute to the placenta or to the embryo itself. In this 8-cell embryo, called a morula for its berry-like appearance, all cells express a pair of genes, CDX2 and OCT3/4. But as the cells of the morula go through their next round of division, CDX2 is downregulated in the innermost cells, while OCT3/4 remains expressed throughout. The segregation of these genes is maintained as the cells continue to divide, until after about 10 rounds of division, the embryo has become a hollow ball, known as a blastocyst. The outer layer of the blastocyst is made up entirely of cells that express CDX2. This trophectodermal layer will go on to become placenta. On the interior, OCT3/4 marks a small cluster of cells with phenomenal ability to differentiate. This inner cell mass is made up of cells with the developmental potential to give rise to all of the body's many cell lineages, an ability known as pluripotency. And indeed, every one of the cells in your own body can trace its ancestry back to these few dozen pluripotent stem cells. The vast majority of the body's trillions of cells are differentiated, meaning they have taken on specialized properties and functions, and have lost the ability to generate cells of other types. Skin, muscle, blood, bone, and the nervous system are all made up of populations of differentiated cells. There is another type of cell, however, that remains less differentiated and retains its ability to give rise to other cell types. These cells are known as stem cells. Stem cells exhibit two characteristics not found in more differentiated cells. By definition, stem cells are able to divide and make copies of themselves indefinitely, a capacity known as self-renewal. This endless power of division is not limited to creating more stem cells, however. They can also give rise to a wide range of mature cell types. These unique characteristics make stem cells indispensable, both for replenishing the body's cells as they age and are lost, and for constructing the body itself from a tiny number of cells during development. Even after the embryo has grown into an adult, many of the body's cells continually die off and need to be replaced. For example, billions of red blood cells need to be replaced each day. This is the work of stem cells known as hematopoietic stem cells, which are located in the bone marrow. Residing in a complex cellular environment, the hematopoietic stem cell is able to generate precursors to every type of blood cell. And indeed, these stem cells have been used by physicians for decades to replenish patients' blood supplies following radiation treatment for cancer. The stepwise differentiation of a stem cell's progeny depends on combinations of genetic and environmental factors that gradually steer these cells into following a specific lineage. This process involves different sets of stimulating factors operating in the cell's environment, at its surface membrane, and within its nucleus. Each step down the path draws it closer to becoming a functional red blood cell and entering the bloodstream. The lining of the small intestine turns over even faster than the blood. At the microscopic level, this epithelial surface consists of billions of projections called villi, with stem cell-containing areas known as crypts near their bases. The intestinal crypt contains slowly dividing stem cells that maintain their own numbers while also producing rapidly dividing progenitors. These transit-amplifying cells churn out all of the differentiated cell types needed to migrate upwards and replace older cells as they are shed from the tips of the villi. This constant self-renewal by stem cells is a feature of many of the body's systems, including skin, hair, and bone. Not all of the body's organs regenerate as readily as the blood and the lining of the gut, and the multipotent stem cells that have been identified in the adult body are both limited in their ability to generate other lineages and difficult to maintain in culture. In contrast, the pluripotent cells of the inner cell mass can be harvested and grown indefinitely in vitro, and can give rise to every type of cell in the body. These pluripotent cells in culture are known as embryonic stem, or ES cells. By culturing these cells under controlled conditions, it is possible to steer their differentiation into specific cell types. For example, ES cells cultured on a bed of stromal feeder cells and treated with inductive factors such as BMP4 and sonic hedgehog tend to differentiate into neural lineages. By testing various ES cell culture conditions, scientists are gaining new insights into the biology of the differentiation processes that build and sustain and may one day be used to heal our bodies. Stem cell researchers are currently exploring ways to differentiate ES cells into lineages that are of great medical and biological interest. The nervous system, the pancreas, and the heart all regenerate too poorly to restore themselves after serious injury or degenerative disease. Scientists are now seeking ways of using the knowledge they gain every day from the study of embryonic and somatic stem cells to help save patients' lives and to improve our understanding of their seemingly limitless potential.
So what we saw in the previous movie, we saw that cells can make the decision to differentiate. In general, when we talk about the cell as a complex system, those decisions that are made by cells are the actuator of the system. So those are the decisions that a cell can make and there's not actually many decisions a cell can make. Three other possibilities for decision making for cells are to proliferate or perform cell division, to die or to form self-execution, that's called apoptosis, and later on we will talk about the cell signaling that induce the process of apoptosis. And a third movie that we're going to just watch is how cells can decide to crawl towards a specific direction based on some signals that the cell receives. So the first movie show you under the microscope the process of cell division.
New cells are formed by cell division, in which one cell splits to become two cells. Cell division involves two processes. In the first, called nuclear division, the nucleus divides. In the second process, cytokinesis, the cytoplasm divides, producing two cells.
In this movie, we'll see cells performing self-execution or apoptosis. This is a famous movie that was filmed many years ago, and it's been watched in YouTube by over 1 million viewers, and it visualizes a macrophage chasing a bacteria. Both the macrophage and the bacteria appear to be alive, and the fact that the macrophage knows the direction where to crawl to follow the bacteria and eventually swallow it is pretty amazing. Now we're going to start talking about molecular components that make up the cell, and one of the most important molecular components that is very relevant to cell signaling networks is the plasma membrane. This is the barrier that I talked about when we talked about agents in general having a barrier. This is the barrier of the human or mouse cell, and it's made of a lipid bilayer that this movie explains in a little bit more detail.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane just 2 molecules thick.
So some of the systems that we're going to discuss is the cell signaling network that takes information from the environment and transduce that information into the cell through cell signaling pathways, such as this MAP kinase pathway that we're just going to watch. And those signals are forming a network and then informing the cell of the status of the environment and helping the cell to make the final phenotypic decision. This MAP kinase pathway, it's probably one of the most famous, well-studied pathways in molecular biology. However, you have to keep in mind that in this animation, the processes are highly simplified, and it seems like there is only one pathway and one cascade that is going at once. However, you have to keep in mind that there are many other proteins and players in this pathway and this process that are not visualized.
MAPK signaling is activated through binding of a growth factor to the extracellular domain of the tyrosine kinase receptor. Signaling molecules GRB2 and SOS are next recruited to the internal docking site, resulting in RAS activation at the membrane. The efficiency and duration of signal transmission is regulated by the scaffolding protein kinase suppressor of RAS, KSR. RAS triggers a phosphorylation cascade involving RAF, MEK and ERK proteins, leading to ERK activation and translocation to the nucleus. Once in the nucleus, ERK activates several transcription factors that mediate gene expression. Target genes thus activated by MAPK signaling contribute to higher proliferation and survival.
Another very important pathway is the PI3 kinase pathway, which is important for making the decision to stay alive.
PI3K signaling is initiated by the growth factor binding to the tyrosine kinase receptor, leading to receptor dimerization. As a consequence, lipid kinase PI3K is recruited to the internal docking site and becomes activated. PI3K next converts membrane lipid PIP2 to its active PIP3 form, which in turn leads to activation of a key signaling kinase, AKT. AKT promotes cell growth through protein synthesis driven by mTOR signaling and reduces cell death by blocking FOXO activity.
And this is the visualization and explanation of two apoptosis pathways, one through an intrinsic signal of DNA damage and the other through an extracellular signal through a TNF receptor.
Two signaling mechanisms exist for activation of apoptosis, an intrinsic mechanism and an extrinsic mechanism. In the intrinsic pathway, cell death may be triggered through DNA damage or severe cell stress. Following activation, intracellular pro-apoptotic proteins are released to activate cascade. This ultimately results in apoptosis, a process commonly blocked in tumor cells. Restoring the intrinsic apoptotic pathway after it has been inactivated may promote tumor cell death. The extrinsic pathway is activated in response to diverse external signals, such as APO2L trail. This pathway has the potential to induce death of tumor cells independently of the intrinsic pathway. Development of novel molecules that promote apoptosis by targeting both pathways of apoptosis, intrinsic and extrinsic, advances the understanding of the mechanisms behind tumor cell proliferation.
So one of the most exciting areas of research now, systems biology and in biology in general, is this concept of epigenetics. We already know the DNA sequence of a human or a mouse cell, but the question remains to understand how those genes that are expressed in our cells are regulated. And epigenetics is the field that tries to understand that at the chromatin level. So the DNA is coiled in chromatin, and that chromatin is regulated by signals from the environment, and this enables the recruitment of proteins that regulate the expression of genes, and the expression of those genes give rise to the cell phenotype. In this particular movie, we're going to look at how the chromatin is organized and how post-translational modifications can lead to the reorganization of the chromatin structure.
The tails of core histones labeled here can be altered with distinct chemical modifications, including methylation of histone H3, acetylation of histone H4, and phosphorylation of histone H2B. Euchromatin is often characterized by a more open and accessible state of the DNA, one in which transcription factors have access to their cognate binding sites, and can therefore recruit enzymes, like histone acetyltransferases, that acetylate histone tails and activate genes by recruiting components of the basal transcriptional machinery, including RNA polymerase. Heterochromatin, in contrast, is thought to be characterized by a more repressive, tight bundling of nucleosomes, which impedes transcription factors from gaining access to regulatory sites on the DNA. Methylation of cytosine bases in regions called CpG islands is a hallmark of transcriptionally repressed heterochromatin. These methylated cytosines in turn recruit proteins like MECP2, methyl CpG binding protein 2, and HP1, heterochromatin protein 1. These proteins are thought to maintain a repressive state of chromatin by inducing histone deacetylation by HDACs, as well as histone tail methylation by histone methyltransferase enzymes.
A very important concept that we're going to discuss in the upcoming lectures is the concept of transcription factors. Transcription factors are proteins that can bind to the DNA directly and regulate the expression of genes.
In the nucleus, DNA is condensed into a DNA-protein complex called chromatin. In places where genes are being expressed, there are often zones of naked DNA. We will now explore ways that transcription factors interact with these zones. Even among scientists, a common misperception is that transcription factors fall from the sky onto the DNA and test one spot at a time. Such a process is too inefficient. Instead, the transcription factor interacts non-specifically with the DNA backbone. This allows it to efficiently search the DNA for its binding site, a DNA sequence it recognizes. The transcription factor slides along until it encounters its binding site. Then it changes conformation to interact specifically with nucleotides on the interior of the helix. Now the transcription factor can stay and interact with other proteins for transcription. Or it can become dislodged by many factors, which could include movement of the DNA or interaction with other proteins. Studies have suggested that transcription factors sometimes move from one part of the DNA to another. Short hops might allow them to bypass other proteins or to hop over compacted sections of DNA. When the transcription factor lands on another segment of DNA, short hops become big jumps. Although the other strand is nearby in three-dimensional space, it may be much farther away on the chromosome or on another chromosome entirely. There are 1,500 different types of transcription factors in human cells. They interact to create a complex language of gene expression.
Once the transcription factors have assembled a complex near genes, transcription can begin, and this movie shows you the process of transcription, a process where a DNA sequence is translated into an mRNA molecule, and then this mRNA molecule will give rise to a protein.
What you are about to see is DNA's most extraordinary secret: how a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
The final movie is about the protein translation process. In this particular movie, we will see how the mRNA molecule is exiting the nucleus after it's been translated, and then through the ribosomes, the mRNA molecule is giving the information needed for the ribosome to assemble a new protein. And those proteins, as you've seen earlier, have many functions, including signaling, but also giving rise to the structure and function of all workings of the cell.
The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for. There can be several million ribosomes in a typical eukaryotic cell. These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the 3-dimensional proteins that are essential for life. Let's see how it works. The ribosome is composed of one large and one small subunit that assemble around the messenger RNA, which then passes through the ribosome like a computer tape. The amino acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules, also referred to as tRNA. The small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes each amino acid and joins it onto the growing protein chain. As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. There are 3 locations inside the ribosome, designated the A site, the P site, and the E site. The addition of each amino acid is a 3-step cycle. First, the tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA. Next, provided there is a correct match, the tRNA is shifted to the P site, and the amino acid it carries is added to the end of the amino acid chain. The mRNA is also ratcheted on 3 nucleotides or 1 codon. Thirdly, the spent tRNA is moved to the E site and then ejected from the ribosome to be recycled. As the protein synthesis proceeds, the finished chain emerges from the ribosome. It folds up into a precise shape determined by the exact order of amino acids. Thus, the central dogma explains how the 4-letter DNA code is, quite literally, turned into flesh and blood.
There is a really amazing video that was created in the 70s by people from Stanford, where they animated the process of protein translation in a dance. So this is it for talking about the biological concepts that you would need in this course. However, I would like to encourage you to continually study the field, and this will help you. If you ever have the opportunity to work with biologists, it would be very important that you can speak their language and understand the problems that they are facing at a high level of detail. Hi, in this lecture I'm going to talk to you about small world and scale-free networks. But before we start, I would like to mention a few things about modeling. So as I said in the first lecture, a model is a theory that is consistent with the data. It's a coarse-grained abstractions of the system. It needs to be cognitively and mathematically understood, so you can trace the variables in the models and you can understand its behavior. It needs to represent the structure and or the dynamics of the real system. It needs to be predictive. And one famous saying by George Box is that models are all wrong, but some are useful. So the application of network modeling of complex systems started in 1736 by Euler, who used the newfound field of graph theory in mathematics to prove the 7 bridges of Königsberg problem. A lot of advancement in graph theory and the application of network analysis in complex system was made by those 2 Hungarian mathematicians, Paul Erdős and Alfred Rényi, in the late 50s. What they did is to study the properties of random networks. So their famous random network model placed some buttons on a table and then connected those buttons with random connections. And they tried to understand the properties of those networks, of those graphs. And one of the properties is that nodes in those graphs have an average connectivity. And that connectivity distribution, if you plot the number of connections per node and the frequency of nodes having a specific number of connections, it follows a Poisson distribution which is similar to the normal distribution. It's a bell-shaped curve distribution. Erdős and Rényi were also famous for proving the problem of how many colors would you need in order to distinctly color states on a map. So in the late 90s, there were two seminal publications that really made a huge impact on this idea of applying network analysis to real-world complex systems. And the first paper was published in Nature by Steve Strogatz and Duncan Watts. And what they showed is that real-world systems follow this small world topology. So what does that mean? And this actually has a very specific mathematical definition. So real-world networks are not random, are not like the Erdos-Renier model networks. They actually have these properties where they have high clustering coefficient and relatively short characteristic path length. So those are two network properties that can be measured: the level of clustering, the average distance between pairs of nodes. So Steven Strogatz and Duncan Watts analyzed three complex systems abstracted to networks. One is the movie actor database, IMDb. They also analyzed a power grid system that lists transformers and generators, and also they analyzed the neuronal connectivity map of the worm C. elegans, where this worm is the only organism that we mapped its entire neuronal connectivity system. Worm has 200 neurons that we know that exactly how they're connecting. So as you see, they compare the actual L, which is the characteristic path length, to the L that is expected for random networks of the same size, as well as the C, which is a clustering coefficient, and compare that to the clustering coefficient expected in random network of the same size. So how do you compute the clustering coefficient and the characteristic path length? So to compute the clustering coefficient, or what the clustering coefficient means, is it's a measure of how the neighbors of a node are connected, the direct neighbor of a node are connected. And in this particular example, you can see that the red node has 4 neighbors. And in the first example, when the clustering coefficient, when C equals 1, all of the neighbors are connected. So there are 6 possible connections that those 4 nodes can be connected, and in the first example on the left, all of them are implemented. In the middle, the C equals half, only 3 out of this possible 6 are implemented, and the clustering coefficient is 0 for that particular node if there are no connections between the neighbors. So you can compute the clustering coefficients of all the nodes in the network, and then the average is the clustering coefficient for the entire network. The characteristic path length is an average of all the shortest path distances between all pairs of nodes. One of the things that Watts and Strogatz did in their paper, they created a model that can generate a network that follows the same small world topology that real world networks also follow. So in this model, they started with a regular lattice and then gradually randomized the connections, taking a link and then putting it somewhere else. So they extracted one of those links and then gradually randomized the network. So if you do that process, starting with this regular lattice, going all the way to random network, somewhere in the middle you're going to get a small world network. So what they've done as they go through this process of randomizing those links from the regular lattice to the completely random network, they can do a measurements of the clustering coefficient and the characteristic path length at every few steps. And what they found is like somewhere in the middle you still are retaining the high clustering while your path length drops very rapidly. And this explains or fits the real world topology. suggesting that there are shortcuts in real-world networks that connect modules that are highly clustered. So in this paper that was published in Science in 1999, Albert and Barabasi noticed is that real-world network connectivity distribution is not like the Poisson distribution that was reported by Erdos and Renyi in 1959. It's actually following a power law distribution, a power law. So the connectivity distribution has a power law density distribution histogram. And this is what is shown here. And then they also, like Watson-Strogatz, they had a model that can generate those power law distributed networks that they called scale-free networks. So let's look at how the model works. Instead of starting with all the nodes and all the links and gradually shuffling the links, the rich get richer model, network growth model that can generate scale-free networks or networks that have a parallel distribution, starts with very few nodes that are randomly connected. So here we have an example of 5 nodes with some random links that connect. Then you start adding nodes and links to the system. So you're introducing nodes, growing the network in size until it reaches your desired size. And that growth model has some basic rules of how the new node that is introduced to the network will be connected to those other nodes. If we are interested in generating an Erdos-Renyi connected network, we will just randomly connect a new node and just grow the network by adding nodes and randomly connect those nodes to the network. However, with the rich get richer model, we are connecting the new incoming nodes by assigning a probability for each node to be connected to the new node based on the node's already existing connectivity degree. One of the things that Barabasi showed is the importance of hub nodes. Most of the nodes in the network have very few connections, either 1 or 2 connections. And then there are those highly connected nodes that, although there are not that many of them compared to the lowly connected nodes, there is a lot more of those hubs compared to the hubs that you can find in an Erdos-Renyi network. And those hubs are important components in the network. If you destroy those hubs, obviously the network will fall apart faster. And what they showed in these 2 papers that were published in Nature is that when you knock down genes that are highly connected in yeast, the viability of the organism is going to be much lower than if you would have knocked down genes that are lowly connected. So in these review articles that Barabasi wrote many years ago, he compared the view of a random network to a scale-free network. One of the criticisms about the scale-free model is that when you look at many complex systems, you see those power law distributions a lot. And this is because complex systems are made of diverse components, diverse agents. And those different types of agents will have different properties. When you're comparing a lot of things that are different, that are diverse, heterogeneous, then you will get those power law distributions. So this is not necessarily, or at least the opponents of the scale-free model suggest that this is not necessarily a feature of the structure of the system by design, but just because we are looking at a very diverse system. If you compare people's height, you will get a bell-shaped curve distribution. However, if you compare all animals' height and weight, you will probably get a power law because you are comparing so many diverse things. So now I'm going to turn my camera around and I'm going to show you what I'm looking at. And this is north part of New York City and Manhattan. And you can see that there are many buildings of many different heights that I'm looking at. And in this particular case, if you would have just plotted the distribution of the height of those buildings, you probably also would obtain a parallel distribution because those buildings are very different. But if you only picked a specific type of building for only the 5-floor buildings, or that you'll see that then you can obtain a normal you can see that you can obtain a bell-shaped curve distribution and not necessarily a parallel distribution. One thing that to keep in mind about the scale-free distribution is that there is sometimes a drop in the hubs. So the hubs, you cannot find infinitely large hubs. So although the power law curve would suggest that there are very large hubs, sometimes we see a drop in the size of hubs. And this can be explained by two mechanisms. So an interesting follow-up paper that came after the publications of Albert and Barabasi suggested an explanation for that drop in the distribution of hubs at the tail of the scale-free distribution. And this, like the building that we just looked at, suggests that there could be some type of a limit of how tall a building can be. They modeled two types of mechanisms that can explain this drop of the tail in the hubs. One is suggesting that the hub nodes age, and as they age, they lose the capacity to accept new links in the rich get richer model. Another one is that there is some type of a physical limit, a maximal physical capacity for a hub to grow, and once that hub reaches that maximal physical capacity, that hub will stop growing. We will talk more later about other mechanisms that can generate scale-free networks.
I don't ever slow up, no I don't take shit. I got no love for the fakeness. If you wanna play tough and wanna hate this, I'll always show up.
I don't ever slow up, no I don't take shit. I got no love for the fakeness. If you wanna play tough and wanna hate this, I'll always show up and make a statement. I don't ever slow up, no I don't take shit.